Good evening, one and all. On behalf of Dr. Agarwal's Institute of Optometry, I extend a warm welcome to each and every one of you to the 18th episode of Optometry Series 2.0. I'm very happy to welcome my team members, Ms. Meenakshi and Ms. Anshika ma'am. Thank you so much for joining with us today. And we have a special guest, Ms. Jemima ma'am pitching in. So Jemima ma'am is known for her case presentation series on YouTube on her own channel. So thank you so much for joining with us, Jemima ma'am.
Thank
And
you.
I'm like to welcome the moderator for today's episode, Mr. Rohit Gupta sir. Sir is a lecturer and coordinator at Department of Optometry for UG, PG and PhD at Mahatma Gandhi Mission School of Biomedical Sciences, MGM Institute of Health Sciences, Navi Mumbai. And he's also a consultant optometrist at MGM Hospital, Navi Mumbai. He has 7 years of academic and clinical experience. His areas of specialty include pediatric eye care, specialty contact lenses, low vision, sports vision. Sir has various awards and achievements to his name. He has recently won Chancellor's Medal for Best Postgraduate in MSc Allied Health Sciences and Heartfulness Youth Ambassador Award 2021 by the International Heartfulness Institute. He has also won Best Faculty of the Year Award 2023 at the third international conference organized by ASCON. Sir has trained in Teachers Training Program for Allied Health Science Professionals, Medical Education Unit, Malavia Mission Teacher Training Program 2024. Sir is also active Board of Studies member and a research guide for both UG and PG students at his organization. Thank you so much for joining with us today, sir.
Thank you so much.
The presenter for today's session is Mr. Arunagiri, a third-year Bachelor of Optometry student at Dr. Agarwal's Institute of Optometry. Mr. Arunagiri, please begin your presentation.
Hello, good afternoon. Good afternoon, everyone. Today we are going to see about anatomy of the retina and visual pathway. Retina is the innermost tunic of the eyeball, and it is a thin, delicate, and transparent membrane. It is the most highly developed tissue of the eye. It appears in pupillage red due to rhodopsin, visual purple of the rods, and the underlying vascular choroid. It extends from optic disc to ora serrata. Grossly, it is divided into two distinct regions, posterior pole and peripheral retina, separated by the retinal equator. Retinal equator is an imaginary line which is considered to lie in line with the exit of four vena verticos. Retinal equator line where four cortex veins exit. Retina posterior to this is known as posterior retina. Posterior pole refers to the area of retina posterior to the retinal equator. The posterior pole of the retina includes two distinct areas, the optic disc and the macula lutea. Gross anatomy of retina. Retina extends from the optic disc to the ora serrata. Its total surface is approximately 266 millimeter square. Thickness and structure vary in different regions. Now, retina color. It appears in purplish red due to the rhodopsins. After that, retina becomes opaque and whitish. Regions of retina. Optic disc, macula lutea, peripheral retina. Optic disc. Optic disc is a pink circular area of the retina. Its diameter is about 1.5 mm. This is the site where optic nerve fibers exit the eye. There is no rods or cones will be there. This is physiological blind spot. Since we have binocular ability, we can't appreciate this normally. Physiological cup. Physiological cup is a central depression within the optic disc. Central retina vessels emerges from this cup only. Cup size varies normally. Enlarged cup indicates glaucoma. Macula lutea. Macula lutea is a yellowish area temporal, which is located to the temporal of the optic disc. Diameter is approximately 5.5 millimeter. It is responsible for central and color vision, rich in cones. Cone cells will be rich in the part. Parts of macula. Macula is divided into fovea, parafovea, and perifovea. Fovea centralis. Central depression of the macula. It is diameter is about 1.5 mm. This is the area of maximum visual acuity. It contains only cones. Fovea. The foveola is the central part of the fovea. Other retinal layers are absent here. Foveola is a avascular zone. It allows the sharpest vision. Peripheral retina. Peripheral retina lies outside the macular area. It is also known as extra-areal periphery. It plays important role in peripheral vision. It divides into near periphery, mid periphery, far periphery, extreme periphery. Ora serrata is the anterior serrated margin of the retina. It marks the junction between retina and ciliary body. Retina is firmly attached at the ora serrata. Peripheral retina degenerations are common near this area. Layers of retina. Retina contain 10 distinct layers. These layers are arranged from outmost, from choroid side to vitreous side. Each layer has a specific structural and functional roles. This is the microscopic view of the retina. Internal limiting membrane, nerve fiber layer, ganglion cell layer, inner plexiform layer, inner nuclear layer, outer plexiform layer, outer nuclear layer, external limiting membrane, photoreceptor cell layers, retinal pigment epithelium. Retinal pigment epithelium. Uh, it is fine mottling due to unequal distribution of pigmentations within individual cells gives a granular appearance. This is a single layer which contains 5 million cells firmly attached to its basement in its basement lamina. Lamina vitrea of it is lamina vitrea of Brecht's membrane. Cobblestone appearance 4.2 million to 6.1 million. Area central is 12 to 18 micrometer with 10 to 14 micrometer height. Ora is 60 micrometer in width. It functions for photoreceptors, renewal and recycling of vitamin A, maintain integrity of subretinal space, transport of nutrients and metabolites, mechanical support to the process of photoreceptors. This is the OCT image. Layers of rods and cones. It contains photoreceptor cells. Rods are responsible for scotopic, dim environment vision. Cones are responsible for photopic vision, daylight and color visions. Outer segments lie close to the retinal pigment epithelium. Cone cells. Cone cells are one of the photoreceptor. It is 40 to 80 micrometer long. They are longest at the fovea and shortest at the periphery. Cones are responsible for eye visual acuity and color vision. Structure of cone. Outer segment is in conical shape. It contains iodopsin pigment. There are approximately 1000 to 1200 lamellar discs per cone. Discs are continuous with the plasma membranes. The inner segment contains a large number of mitochondria. Cone ellipsoid is plump. Cone nucleus lies in the outer nuclear layer. Cone pedicle forms synapses in the outer plexiform layer. Rod cells. Rod cells is 40 to 60 micrometer in size. Cylindrical, highly retractile, transversely striated, and contains visual pupil. Has rod spherules. Rod cells has rod spherules. Visual pigments. Rods. Rods are helping for scotopic vision, greatest sensitivity for blue-green light. Visual purple are also called as rhodopsin. Cones, trichromatic pigments, short blue 440 nanometers, and medium green 540 nanometers, long orange 577 nanometers. External limiting membrane. It appears as a thin fenestrated membrane formed by junctions between the photoreceptors layer and the Muller cells. It is not a true basement membrane. It functions as a act as a selective barrier for nutrients, and it is helping in stabilization of position of the transduction portion of photoreceptors. And outer nuclear layer. It contains soma and nuclei of photoreceptor cells with varies nasal to disc 445 micrometer to 8 to 9 rows of nuclei. Temporal to disc 22 micrometers, 4 rows of nuclei. Fovea, 50 micrometers, 10 rows of 10 rows. Different nuclei can be differentiated because nuclei stains with Mallory stain. Outer plexiform layer. It synapses between rod spherules and cone pedicles with dendrites of bipolar cells and process of horizontal cells. It marks the junction of end organs of vision and second order neurons in the retina. Thickest at the macula, 51 micrometers. Micrometers consist of predominantly of oblique fibers that have deviated from fovea, also known as Enel's layer. Inner nuclear layer. It's a very thin layer, disappears at fovea. It consists of bipolar cells, horizontal cells, amacrine cells, soma of Muller cells, capillaries of central retinal arteries. Bipolar cells. They are the first order neurons of vision. They cannot they connect photoreceptors to ganglion cell. Several structural types are present. There is this explains the types, connections, and the features of bipolar cells. Rod bipolar cells is 20% large soma profuse dendrites. The connections are arborized only with rod spherules. Its features are axons of these bipolar cells have synapse with soma up to 4 ganglion cells. Midget bipolar cells make connections only in triads of cone pedicle. Its feature is invaginating deeply invaginate cone pedicles. Flat makes superficial contact with the cone pedicle. Axon synapses with single ganglion layers. Diffuse. It connects it contact with the cone pedicles only, not with their triads. Axons synapses with a number of ganglion cells of all types. Blue cone bipolar cells. Innervate more than one cone pedicle. Giant bipolar cells distinguished by extent of their dendritic spread. Horizontal and amacrine cells. Horizontal cells provide lateral inhibition. They help in contrast enhancement. Amacrine cells perform integration and modulation of signals. Muller cells are the main glial cells of the retina. They provide structural and metabolic support. They extend from external limiting membrane to internal limiting membrane. Inner plexiform layer. This layer contains synapses between bipolar and ganglion cells. Amacrine cells processes also present here. It is absent at the foveola. Ganglion cell layer mainly composed of cell bodies third order cells. Other process of Muller cells, other neuroglia, branches of retinal vessels are present. Single layers situated at peripheral retina, two layers temporal side of optic disc, 6 to 8 layers edge of the foveola. 1.2 million ganglion cells present in retina. Each produces single axon. It converges and exits from from the eye as optic nerve. Can be classified according to size, degree of arborization, spread of their dendrites, pattern of synaptic connections with amacrine and bipolar cells. There are two major types, M cells and P cells. M cells project the magnocellular layer of lateral geniculate body and exhibit non-opponent response. P1 midget cells contribute 90% of total ganglion cell layer at foveola. P2 small by stratified. Nerve fiber layer. Unmyelinated axons of ganglion cells will be present. Converge at optic nerve head. Pass through lamina cribrosa. It becomes ensheeted by myelin posterior to lamina cribrosa. Arrangement of nerve fiber in retina. Nasal half directly to optic disc as superior and inferior radiating fibers. Macular region pass straight in temporal part of a disc as papillomacular bundle. Temporal retina arc above and below the macular papillomacular bundle as superior or inferior accurate fibers. Arrangement in optic nerve. Fibers from peripheral part deep in retina occupy most peripheral part of optic disc. Fibers closer to the optic disc superficially in retina and occupy more central deep portion of Its internal limiting membrane. It is a true basement membrane. It separates the retina from the vitreous body. It is formed by Muller cell foot plates. Now, blood supply of the retina. Central retinal artery supplies to the inner retinal layers, inner nuclear layer, inner plexiform layer, ganglion cell layer, nerve fiber layer. This is the branch of ophthalmic artery. Chorea capillaries are originated from short posterior ciliary arteries. It's a branch of ophthalmic artery. It supplies to retinal pigment epithelium, photoreceptors, external limiting membrane, outer nuclear layer. Blood retinal barrier is formed by tight junctions of retinal capillary endothelium, retina pigment epithelium, prevent leakage of plasma, maintains retinal homeostasis, breakdown seen in diabetic retinopathy. Now, visual pathway. Each eyeball acts as a biological camera, converts light into neural signals. Visual pathway is helping to reach the signals to the visual cortex. Optic nerve. It is a second cranial nerve. Extends from the optic disc to optic chiasma. Backward continuation of retinal nerve fiber. It contains 1 to 2 million axons. Axons are arising from retinal ganglion cells. It contains afferent fibers for light reflex. It contains few centrifugal fibers. Characteristics of optic nerve. It is an outgrowth of brain tissue comparable to white matter tract. Not covered by neuralema. No regeneration after injury. Fiber diameter 2 to 10 micrometers. Peripheral sensory nerves almost 20 micrometers. Surrounded by meninges. Primary and secondary neurons located in retina. Parts of optic nerve. Totally 47 to 50 millimeter in length. In intraocular part, almost 1 mm. In intraorbital part, it almost 30 mm. In intracranial part, it is 6 to 9 mm. Sorry, in intracanalicular part, it is 6 to 9 mm, and in intracranial part, it is almost 10 mm. Intraocular part, optic nerve. It lengths around 1 mm, begins at the optic disc. Retinal fibers make orthogonal turn. Fibers passes through lamina cribrosa. Emerges as intraorbital optic nerve. Diameter is around 1.5 mm. Diameter behind sclera is almost 3 millimeters. Structure of optic nerve. It divided into 4 regions. Surface nerve fiber layer, T-laminal region, lamina cribrosa, retrolaminar region. Surface nerve fiber layer composed of retinal nerve fibers, 94 percentage, astrocytes 5 percentage. It's covered by internal limiting membrane of L-sling, separates optic disc from the vitreous body. It's the central thickening called central meniscus of Kunt. Other retinal layers separated by intermediary tissue of Kunt. Pyramidal region, increased number of neurons, abundant astroglial tissue, border tissue of Jacobi present. Astrocytic cuff separates nerve from choroid. Lamina cribrosa. It's a sieve-like structure made of fenestrated scleral connective tissues, lined by glial tissues. It bridges posterior scleral canal. Optic nerve fiber pass through uh, fenestration. This picture is disturbing. Border tissue of Elschling present rich blood supply from circle of Zinn retrolaminar region, decreased astrocytes, acquisition of myelin sheath, myelination by oligodendrocytes, diameter increases from 1.5 mm to 3 mm, axonal bundle separated by connective tissues, septa, posterior limit not clearly defined. Intraorbital part, it lengths around 25 to 30 millimeters, extends from the globe to optic canal, longer than straight orbital distance, S-shaped course, allows free eye movements, covered by dura, arachnoid and pia. Subarachnoid space contains CSF. Relations intraorbital part, central retinal artery and vein enter inferomedially about 10 mm behind globe. Anterior relation, orbital fat, extraocular muscle separator, posterior relation, annulus of Zinn, and the origin of four rectus muscles. Adherence of superior and medial rectus fibers. It causes pain in retrobulbar neuritis. Additional orbital relations, long and short ciliary nerves, long and short ciliary arteries, ciliary ganglion, oculomotor nerve division, nasociliary nerve, sympathetic fibers, abducens nerve, ophthalmic artery crosses superiorly. Intracanalicular part lies within optic canal, closely related to ophthalmic artery. Ophthalmic artery crosses inferiorly. Medial relations, sphenoid sinus, posterior ethmoidal sinus, thin bony separation, explain neuritis in sinus infection. Intracanalicular part, it length around almost 1 centimeter, lies above cavernous sinus, converges to form optic chiasma. Its diameter is 4.5 mm, covered initially by pia mater only. Pia mater receives dura and arachnoid at optic canal. Relations cranial part, internal carotid artery, inferior initially, lateral subsequently. Ophthalmic arteries arises below nerve. Structures above anterior cerebral artery, olfactory tract root, anterior perforated substance. Meningeal sheath of optic nerve, intracranial part, pia mater only, intraorbital and intracanicular part, pia arachnoid dura. It is sheath continues with the brain meninges. Subarachnoid space contains CSF, anteriorly fused with sclera. Dura splits at orbital apex. Pia sends septa into nerve. Pia plus glial tissue is called pia glia. Optic chiasma. It is flattened in structure. Width is around almost 12 mm. AP diameter is 8 mm. Lies over diaphragma sella, surrounded by CSF. Forms anterior wall of third ventricle. Nasal retinal fibers cross almost 53 percentage. Variations of optic chiasma. Central chiasma 80 percentage above sella, affected early in pituitary tumors. Prefixed chiasma is 10 percentage, lies anteriorly. Optic tracts affected first. Um, Post-fixed chiasma 10 percentage, lies posteriorly. Optic nerves affected first. Relations of optic nerve, optic chiasma. Anteriorly, anterior cerebral arteries, posteriorly pituitary stalk, mammillary bodies, superiorly third ventricle, inferiorly pituitary gland, laterally internal carotid artery. Optic tracts arises from posterior lateral chiasma, run backward and laterally. Each tract contains temporal retinal fibers, nasal retinal fibers, temporal retinal fibers of same eye, and nasal retinal fibers of opposite eye. Terminate in lateral geniculate body. Pupillary fibers pass to superior colliculi. Lateral geniculate body. Oval gray matter structure. It's located at termination of optic tract. It's composed of 6 neuronal layers. Alternating gray and white matter acts as a relay station for the vision. Optic radiation. It is extend from the LGB to visual cortex, passes through retinolenticular internal capsule, form fan-shaped medullary optic lamina. Inferior fibers, Meyers's lobe, temporal lobe in upper visual field, superior fibers in parietal lobe, lower visual field. Visual cortex location, medial surface of a occipital lobe, around calcarine fissure, may extend laterally, limited by sulcus lunatus. Functional visual areas, V1 area 17 primary visual cortex, V2 area 18 secondary visual cortex, V3 anterior part of area 18, V4 area 19 color and form, V5 responsible for motor perception. Blood supply of visual pathway. Optic nerve. Um, for optic nerve, prelaminar and laminar. They are short posterior ciliary arteries are supplying to this circle of Zinn-Haller. For retrolaminar and intraorbital part, pial vascular plexus, branches of ophthalmic arteries are supplying. For intracanalicular and intracanal part, ophthalmic arteries, internal carotid artery branches are supplying. Optic chiasma, anterior cerebral artery, arterial communicating artery, posterior communicating artery, internal carotid artery. For optic tract, anterior choroidal artery, posterior communicating artery, posterior cerebral artery. Visual cortex, area 17. Posterior cerebral, the blood supplies are posterior cerebral artery, calcarine artery, dual vascular supply, macula sparing. Conclusion. The retina converts light into neural signals. This signal travel through a well-organized visual pathway to the visual cortex. Partial crossing at the optic chiasma enables binocular vision. Lesions at different levels produce specific visual field defects. Knowledge of this anatomy is essential for clinical diagnosis and management in ophthalmology. Thank you.
Thank you so much, Arun. It was a very elaborate and extensive presentation on retina and visual pathway. As of now, we don't have any questions on the anatomy part for the moderator. Anshi ma'am, from your side, do you have any questions for our moderator, ma'am?
Hi, Meenakshi. Rohit, sir, can you give an outline on what should the students focus or how should they try to remember the anatomy and visual pathway? Because what happens is when they go to the second year or third year, it becomes very difficult. So, what could really help them to remember and how actually the retina works and how anatomy actually plays a very important role in understanding the diseases and the parts which are affected?
Am I audible? Hello.
Yes, sir. Yes, sir.
Now you're audible, sir.
Yeah, I'm audible. Yeah. Okay. Actually, a very genuine question, and I guess maximum of the teachers face this issue with the students. Like in the second year, when we teach them about the retina, how it's going to work and how it is important. But rather than learning, they are afraid about it. That we have to by heart the layers of the retina. Another part is understanding the structures. Okay, now the first time when they see the image, it's very fascinating for them. The main thing is where to look at and what to look at. As in the presentation, a beautiful presentation, first of all, very descriptive. And third year student presenting such a broad spectrum about retina, and that is really, applaudable, that I should applaud for him. Yeah, I'll answer this. I have written certain things in which students face the most difficulty. The first is the layers of the retina, how to, instead of by hearting, I feel they need to understand how these things work. I'll first focus on the macula. When we say macula, we just only focus on that is the central part where the most of the image will get focused. You'll get the clarity and everything. But when we are talking about the anatomy and understanding its physiology also and its working principle, we need to go beyond certain things, such as why there is this dark color. Okay, why we see that it is more darker. Okay, the answer is it is highly pigmented. When I say it is highly pigmented, it means there are these tall epithelial cells of the RP, but which gives them the darker color. But also we need to understand that it is the dense pigmentation which will help them reduce the scattering of the light. Now, this is how they can start understanding. In spite of only looking at it, how we will understand highly pigmented epithelium or else, different layers and what is this function. Now, understand why there is this layer or else why there is the central part, why it is darker and how it will help and why the darker color is there. If I say it is highly avascular or else it is vascular, now you need to understand what is exactly it is happening. If I say that it has also this yellowish UA, so this yellowish UA is to highest concentration, and that is the reason that there is the presence of xanthophyll pigments. Now, for the students, how we can understand this xanthophyll pigments? What are these xanthophyll pigments, and what are they doing on the macula? It is very easy. Now we just go back to the physical optics or geometrical optics or visual optics. We can understand it acts as a filter. It absorbs the short wavelengths. Another part is it will help to reduce the chromatic aberration. So this is how you can break those things into three parts. And now boom, you get to understand what macula is. Okay, it is highly pigmented. It has tall epithelial cells of RPE. Then it gives the darker color. It is dense in pigmentation. It has these xanthophyll pigments present, and xanthophyll pigments will help them reduce the chromatic aberration. Now, another point which I have always seen that has been missed, and now that gets questioned during the internship or as while looking at the OCT report or anything, that why there is this dip in the fovea. If I'm not wrong, there is this confusion that okay, this dip is there, okay, so this is the foveal region, but we need to understand why there is this foveal region present and how that dip is forming. It is very simple. Now, again, we will break those things into, we will differentiate. Now, the macular part is done. Now, we'll focus onto the fovea. Now, fovea, we need to understand we are focusing onto the central part. Okay. In that, we will understand the central depression. So, when I say central depression of the fovea is formed, why? Because there is displacement. Now, what is the displacement that is happening? The displacement is happening of the retinal neurons, which are leaving, and they are leaving on the side and leaving the photoreceptors exposed. That is the reason why you are getting the sharpest visual acuity in the central part. And that is the only reason why we have the sharper vision. We have more of the photoreceptors. That is, obviously, we have cones in the center in the, higher numbers to get more sharper and clearer image. Now, as in the slide said that Henle's fiber. Now, how you can understand this Henle's fiber? That is the photoreceptor fibers, they become larger. When they get displaced, they become larger in space and they get elongated. And these elongation creates the fibers which are called as Henle's fiber. Okay. And another is it is avascular when we say, so there is absence of this blood vessels over there, and that is how it has been created. Now, coming to the another part, jumping to the next layer, the sixth layer, see how I'm dividing. I'm starting to focus on one part of the retina, and then I'm jumping onto the retinal layers. Or else you just first go through the layers of the retina and now just focus on one part, just not reading by the books, but also explore certain, websites or else some papers or else some, uh, some theory which has been written, which is very fascinating and easy to understand. And that will create a gap understanding of okay, we just don't have to read it for the exams, we just need to understand how this thing works. Because I feel that what we should focus as an optometrist is beyond that. Like what the image is going to form. If I'm looking into the speciality, I'll focus on okay, the image is falling or not falling. If there's a squint, center image गिर रहा है या नहीं गिर रहा है, side में गिर रहा है, कैसे हो रहा है, पीछे image proper पहुंच रही है कि नहीं पहुंच रही है. That whole visual pathway. Now the retina will you know connect the visual pathway, and then we will try to understand the visual pathway part. I hope I'm audible.
Yes, sir, you're audible, sir.
Yeah. Please let me know if it is extending. I have just written certain points which I'm trying to connect it. Is
No,
it
no, please
okay
go
with the
ahead,
going with
sir.
the slide?
It clearly is very useful, so please go ahead, sir.
Okay. Yeah. Thank you so much. Another thing which he has beautifully mentioned about the optic disc. When I saw that the diagram and everything, he also mentioned about the peripheral retina, mid periphery. I was like when I made those presentations, it was at the initial phase it was difficult to make understand, and he understood very well. But now again, we need to understand that that is the landmark. The optic disc is an important landmark of the retina. It is not just the part from where everything is going away. Actually, it is taking the lot many informations from that area to the end of the the brain. Now we talk about V1, V2. First, we need to understand how this image is getting carried forward. So in the part where we are also focusing on the optic disc, also focus upon the around the optic disc what is present. There is a neuroretinal ring. So this neuroretinal rim will help in another things. That is, it has sharp peripheral margins. Why these peripheral margins are present, and then we would focus on this retinal pigments. Are they going to really help it, or else it's just the image transferring part present? I guess this is the I guess that this was the presentation shared to me, and from which I'm just trying to collect. Yeah, the blind spot. Important thing about the blind spot when I said so there is no photoreceptor cells. Now we are jumping from center. We are going to the optic disc part. that there is a physiological blind spot. First, they need to actually explore about the physiological spot. So, where is this physiological blind spot? In my UG period, I learned about this from a teacher, my mentor, that take a thumb and then just rotate it and just see at which particular area. Close one eye and then rotate it into the direction and see at which point you will see that thumb is not being seen. And that is one of the fascinating thing that you get to know, Ariha, this is the blind spot and how it gets covered by the binocular vision. And this is how it works. I guess more practical, more better you try to understand things. Next part is about the amacrine cells, bipolar cells, and the horizontal cells. Now if we look at the image, it's very, we are looking at the layers, all the layers. Now we have learned it. We have gone to the nerve fiber layer, then we are going to the ganglion cell layer, inner lexicon, everything. But on the other side, we have this amacrine cells, horizontal cells, bipolar cells, and then we have this ganglion cells. Now, apart from this, why they are present, what is their main role that we need to understand? So, in that, if we talk about bipolar cells, uh, photoreceptor signals, they forward photoreceptor signals. Then another is horizontal cells. It is onto the outer plexiform. Yeah, outer plexiform. That is the inhibition of the contrast. It works on the contrast. Then amacrine cells works onto the motion and timing modulation. So this is how, instead of just reading about the topic, that is okay, amacrine cell is present, bipolar cell is present, understand that why it is present and what will be its role. Now, why I am briefing on this particular part, that it will work onto the contrast, it will work onto the motion, it will work on another things, because when we read about LGN, that is lateral geniculate nucleus, at that point we read about the three pathways. That's one M pathway, magnocellular, parvocellular, and the coniocellular. And it is my favorite topic in this one, which really helps us a lot to understand how we understand motions, how we capture, how we look at an object. Are we sensing colors? Which pathway is responsible for looking colors? Okay, which pathway is responsible for understanding the movement? All these things are important. As we understand, let's divide this again. Now, magnocellular, parvocellular, and the coniocellular pathway. Yeah, that is there. So, if we talk about magnocellular, it is for the motion. It is for motion depth perception. Parvocellular works on the form perceptions and it looks at the red and green color vision. And when we talk about the coniocellular, it looks on the color contrast and also works on the blue and yellow color. So now understand how these are going to function. So one is for motion, one is for form perception, contrast, fine visual acuity, and then we talk about the blue and yellow and the color contrast. So this is what I suggest to read about. Just get into it so that you get more interest on why these things are present instead of just looking at the slide and dreaming about that these words are difficult. How will we by heart and how will we understand? I guess that is what it is.
Thank you so much, Rohit sir. That was really informative on how making connection makes things easier. So, as you very rightly pointed out, if we just look at anatomy and try to remember it, it's going to be difficult. So, we can try to put in along with the physiology and with the uses and the clinical aspect. So, all together, it becomes like a complete wholesome meal for us to remember and for the brain to also process it. So, thank you so
Yeah.
much, Rohit sir. Gomati, anything else from your side? I think Meenakshi is not there. Yeah. Anything else from your side, Gumati?
So there's this just small point about the visual pathway. The another stem that comes in is understanding the afferent and efferent pathway. When we go along with it, I would always suggest that just don't read visual pathway. Immediately read the afferent and efferent pathway, break it at the pretectal nucleus, continue with the Edinger-Westphal nucleus. I love that name especially. And that eventually that will build up the whole thing and you will just try to sum up everything in one cup, just ma'am said right now. And the whole process when we break it into certain divisions, that is just understanding and just reading part, now it becomes very difficult. There is one word always written onto the textbooks that I remember in AK Kuna, it was written back long ago. Now we don't refer, but yeah, at those times it was really helpful. Sympathetic and parasympathetic. Now looking at those words, now they're like why there it is present over there. Just understand one is for mydriasis, meiosis, one is for mydriasis. Now this is how bring up all those basics and just put it into the one plate. And that is how I would just end up with the pathway. Thank you.
Thank you, sir. Jemima ma'am, would you like to add any words, ma'am?
Yeah,
And
so myself, I take ocular physiology and it is so close to my heart. And I can see the passion that you have in teaching this. It's really nice, amazing to see the passion that you have. And
Thank you, ma'am.
I wish that every student will have the same passion because unfortunately they learn these in their first years and as they move on to the second, third, and when they go to internship, they
Yes.
sometimes they tend to forget. And these sessions are so useful for them because in this platform that we are covering so many topics, and this is available for any student or any faculty to use it. And I'm sure that for Retina, I will ask all my students to refer this optometry series episode. So thank you so much. You took it so well.
Thank you so
And
much, ma'am.
also
It's
thanks
so
for
nice to see you back, after so many, like we have been connected via, chats and everything, met
exactly.
through last time in the ASCON,
Yeah.
and yeah, seeing you here, it's good.
Yeah, happy to connect. Yeah, thank you so much. So I've been asking Rohit and speaking to him about the internship as well. So we do have a lot of interns joining us all over India from various colleges. And soon I hope to have interns from your college also, Rohit.
Sure, sure, ma'am. We'll definitely work on it, and I have promised you sooner or later that yeah, we'll have this connection bond.
Okay, yeah, sure. So even for our internship, we have every Friday, we have classes, sessions where
Okay.
each topic is covered every week. So I will definitely call you for that as well.
Thank you, ma'am.
Okay, then
My
yeah.
pleasure. Thank
Thank
you so
you.
much, everyone, for inviting me. Thank you for connecting.
yes, thank you so much, Rohit sir, for joining with us today. Thank you, sir.
Thank you, ma'am. Thank you.
Thank you,
Thank you.
Jemima ma'am. Yes,
Thank
thank you,
you.
Anshika ma'am, and thank you, Meenakshi, for joining with us today. If you have more questions, you can put it in the comment box. We'll get back to you with the answers. The announcement for this particular week, the admissions are open for Pan India internship and fellowship in optometry at Dr. Agarwal's Institute of Optometry. If you like this video, give a thumbs up and share with all your friends. We meet you in the next episode. Thank you all. Thank you, Rohit sir.